Hello everyone, I'm Dr. Mark Hyman here at the Center for Functional Medicine in Cleveland and at Cleveland Clinic, and I'm so happy to have Dr. Suzanne Goh here, who's an extraordinary scientist, researcher, clinician, whose pioneering work in autism is really changing our thinking about how to approach these children with this devastating disease that's focused not only on behavioral aspects but also on the neurobiology and biochemistry and physiology, and looking really more at autism as a metabolic encephalopathy. And you know, it's sort of really great to have you here at Cleveland, and you just did grand rounds and sort of open up a doorway to a new way of thinking, which is a major paradigm shift, right? From refrigerator mothers to all of a sudden metabolic disorder, which is a very different view. And you know, I'm curious to maybe start off, how did you sort of come as a pediatric neurologist to understand that the old paradigm was broken and that we need a new way of thinking that broadened our view of this disorder?
So I think of it, when I look back at sort of my own path and what's supported and enabled the work that we're doing now, I think it very much started from having a view of medicine from the outside initially. So when I was an undergraduate, I studied, my major was the history of science, which is kind of a unique major.
It's like a liberal arts science degree.
Yes. And fortunate that it was available and that it allowed for also the pre-medical path to be interwoven to that. So that made a big difference, and so I've always had an interest in the medical humanities, which I think have so much to contribute to how we practice medicine. And I also think that I had the opportunity to study, you know, I think cross-disciplinary, interdisciplinary studies are so important, and it really influences the kinds of questions we ask, the research questions, in fact, you know, how we go about research. And so I would say that that was, for me, very formative.
So you had a quote in your lecture from E.O. Wilson, who's a sociobiologist from Harvard, who's written a book called Consilience, The Unity of Knowledge, which is really about this sort of false architecture of medicine and science in general and all knowledge in general, that everything's connected, and that we tend to ignore those connections, and that this artifacts that we have of specialties and diseases doesn't really make sense given what we know now in 2017 about the nature of systems thinking and biology. So I want to sort of dive into a particular window that you've opened up, which is sort of the role of mitochondrial dysfunction in autism, and then not only as a sort of a causative factor, but also a doorway to therapy, which is kind of unusual, right?
Yes.
So how did you come to that, and can you share with us some of the key findings, and then I want to get into how you assess these patients more in detail, and then the treatment, because I think everybody's listening is probably very curious about, well, how do you diagnose it? How do you treat it?
Right. So probably the first thing worth saying is that mitochondrial dysfunction in autism is heterogeneous. So it's certainly not a single entity. The function of mitochondria is so complex. They're probably best characterized as a metabolic signaling platform. So the metabolic pathways and processes that they impact are vast. So mitochondrial dysfunction is not one thing. Even for those with autism, there's some research supporting what seems to be a deficiency or reduced function of certain aspects of the mitochondrial respiratory chain, but there's also research supporting the idea that there's excessive hyperfunction of other elements of the respiratory chain in some individuals with autism, and that maybe one sort of mechanism corresponds more to a phenotype of low arousal, low tone, low energy, and the other
you see a lot of these kids with hypotonia, right?
Yes. And then in contrast, there's another phenotype of hyperactivity, excess energy. And so it's not yet known, but it's possible that there's sort of a correlation between the mechanism and the phenotype, and that mitochondrial dysfunction in autism can look a lot of different ways. But there are some classic presentations, one of which is regression in the first few years of life in the setting of a physiological stressor. There's some very interesting research happening at UCSD in the lab of Bob Navio, looking at what he's termed the cell danger response, which is another way in which mitochondrial function can be disturbed, in which the mitochondria are in a state of essentially sort of a stress state, and it leads to a whole cascade of metabolic disturbances. And there may be some therapeutic interventions that are investigational at this time.
Like the Surman trial.
That's right.
Which is sort of an old drug that was used in Africa for infectious disease, right?
Right.
And now it's being used in autism.
Yes.
And you've seen some interesting positive results around it.
Yes, well, there's just recently we published the first pilot, a very small randomized controlled trial to look at the safety and tolerability of low-dose Surman. And the initial results are positive, and there are plans in place for a second trial.
And the mechanism is around the cell danger response?
That's right. It essentially blocks receptors, purinergic receptors on the surface of the cells that are activated by extracellular ATP. So that the cell danger response, part of it involves the release from cells of ATP. So we know that ATP inside cells is helpful. ATP outside of cells signals this sort of stress state and perpetuates the stress state, which is characterized by increased free radicals and many other metabolic disturbances.
So you see it's almost as an energy deficit in the brain, so that it can't do the normal processing that it does, right?
Right.
Yeah.
So that's thought to be one of the mechanisms.
This is like an acquired mitochondriopathy. It's not necessarily something they're born with, it's an inherited metabolic disorder.
Right. In fact, researchers have looked at, in those children with autism who have biochemical evidence for mitochondrial dysfunction, do they have a genetic abnormality to explain it? And the vast majority do not. So it's thought to be secondary mitochondrial dysfunction rather than primary. Primary being related to a gene defect. The gene defect doesn't seem to be present. So then it raises the question, well, perhaps mitochondrial disturbance is a downstream consequence of other factors, potentially environmental factors.
So environmental toxins.
So exposure to environmental toxins, which, as I mentioned in the presentation, we actually know quite a lot about. We know many of the industrial chemicals that have developmental neurotoxicity, and we know that the mechanism of action of many of them is in impacting the function of mitochondria.
Right. Okay.
So that's certainly one pathway that's increasingly well characterized. And there are contributing factors from inflammation and in general oxidative stress, which mitochondrial dysfunction leads to increased oxidative stress, and it's a
vicious cycle.
Yeah.
Fascinating. Sort of in our work in functional medicine, there's a group of folks that work in the autism space, and the diagnostics we do are pretty on the fringe. We look at stool testing, we'll look at organic acids, we'll look at oxidative stress markers, AOHTG and lipid peroxides. We'll look at nutritional status. And, we often see really extreme abnormalities in these kids. It's like I'm a family doc, so I see babies all the way to like 100-year-old people, and it's fascinating to me that of the thousands and thousands of patients that I've seen, the autistic kids are like really outliers in terms of the degree of abnormalities. They don't have a little oxidative stress; they have a lot, or like test ranges that I've just like in the top 1 or 2%. And do you see that kind of thing where they're just sort of?
Yes, we do. I'll say probably one way in which our practice is a little bit different from maybe a functional medicine practitioner, is that we, in part because we do such a wide range of therapies, that we don't do the degree of some of these diagnostic tests. Also because in young children, it's just harder very often to do the test, and because we're almost always working in a case of some limitation of resources. So, and there are some tests that are, especially for young children, there are things to be ruled out neurologically, like subclinical seizures, status epilepticus of sleep, and so it's a process. But I would very much like for our practice to be doing more of this over time.
If finances were not an issue and it was reimbursed and it was like part of the practice, it would be. So what are you testing? So we look at organic acids, but you measure pyruvate, lactate, alanine, ammonia, as other markers of mitochondrial dysfunction.
So a very routine panel for us would be a CBC, a Chem 20, lactate, pyruvate, plasma amino acids, urine organic acids, and acylcarnitine profile. And that's a very good place to start. Others that we would like to test for if we can get enough blood to do so would be CoQ10 level, selenium, vitamin E level. Others that are part of our very routine initial panels would be thyroid studies, iron, zinc, vitamin D, B12. Yeah.
So take us through sort of what we would find, and then what you do for what were the pearls around organic acids or lactate.
Sure.
Like what are the for a clinician listening, curious, like how do I apply this?
Sure. So some of the patterns that we see most commonly are a mild elevation of lactate. In the setting of a blood draw that wasn't traumatic, the tourniquet was released, the sample was processed properly, and yet lactate is elevated just slightly above the normal range. So not a dramatic elevation, but a mild one. A lactate to pyruvate ratio of greater than 20. An alanine to lysine ratio of 2.5 or greater. Mild elevations of the liver enzymes. Mild elevation of ammonia in a sample that was processed properly. Acylcarnitine profile with very mild elevations across multiple species, different length chain fatty acids.
Profile, yeah.
Or total and free carnitine levels, so a low total carnitine level, low CoQ10. Those are all common findings.
So I understand like the elevations in lactate, because that's indicating sort of mitochondrial dysfunction.
Yes.
But the sort of ammonia and the things like CoQ10 and carnitine, those are those the dietary stuff related to CoQ10 and carnitine? The ammonia, is that related to gut function? Like where's that coming from?
Well, classically we think of these mild elevations in ammonia as being related to liver dysfunction, as well as the elevated transaminases.
That's what we check in hepatic encephalopathy, right? So.
And elevations of ammonia may point to urea cycle disorders if it's higher, but more mild elevations are likely pointing to mitochondrial dysfunction. In the urine organic acids, mild elevations of Krebs cycle intermediates is also another sign. And you're right that there can be multiple sources. So abnormalities in a single analyte could be from multiple different causes, but it really is the overall pattern, that's suggestive.
So you're looking for mild deviations from normal.
Yes.
That indicates sort of subtle metabolic derangement. Yeah.
And in the setting of a suggestive clinical history, it really warrants, then therapy, which can also be informed by the test findings. A very, sort of a mitochondrial cocktail that we commonly use is one that includes L-carnitine, especially if carnitine levels are low, coenzyme Q10, B-complex vitamin, vitamin C, and vitamin E. So that is a very common starting cocktail for us.
And so when you apply these therapies, what do you see in these kids? Because you treat a whole range and often very more extreme cases.
So a wide range of changes. There can be improvements in sort of attention, awareness, self-regulation. Those very often occur sort of early in treatment. A reduction of repetitive and sort of obsessive compulsive types of behaviors. Increase in language. So those are some of the more interest and awareness of social interaction and engagement in social.
Do you ever see kids just wake up and like? Or is it when you combine all the therapies together, like the diet and the gut treatment?
There's a whole range, and for the most part, the improvement is one that's gradual. So less so maybe an awakening, but more one of gradual and increasing sort of developmental progress, across many different areas.
Fascinating. So, one of the things we often think about in functional medicine is upstream causes, right? So mitochondrial dysfunction could be primary, but it's mostly secondary, as you sort of mentioned. So then the question is, how do you go up the chain? Because you can give them mitochondrial support, but unless you deal with the drivers, which is environmental toxins, sources of inflammation, and oxidative stress, how do you think and deal with that?
I think the best approach is to really target the upstream causative factors as well as the sort of downstream impairment. And so, nutritional supplements are helpful, but most helpful when in the context of different nutritional and lifestyle modifications. So there are stressors in the home environment, both related to exposures, so environmental type of stressors, pesticides in foods, or something like EMF, or a variety of factors in the home that may be increasing the burden or the load on a child's metabolism. And then there are also a whole, because so much of what we do is on the behavioral therapeutic side, a whole host of stressors involved in relationships and social interactions that can also be diminished, which has benefit for metabolism.
Right.
So the link.
There's different doorways into it. Yeah, it's fascinating. The things that we find like that are common factors in autism that I think you're probably seeing as well is severe gut dysfunction as a source of inflammation, oxidative stress, food reactions, gluten, dairy, leaky gut, fungal overgrowth, which is sort of like really an outlier in medicine, is some weird thing that we do, but it actually we find it extreme. I mean, in terms of interventions, antifungals are powerful for these kids. Heavy metals, and then this whole methylation sulfation cycle disruption. Do you find the same things in these kids?
We do. I would say probably, a lot depends on the age and sort of the unique presentation of the child. And we go about sort of trying to prioritize and really target potentially the highest yield symptom, pathway
go for
symptoms.
The 80-20 on it.
Yeah, we try to do that, and also are guided by the tests and interventions that, at least in our field, have the most research to support them. So it's always a balance. And as we get through some of those early ones, then we can look in a little bit more detail at some of the other areas that are also hugely important, a little bit outside of my expertise, but which I've made efforts to become educated in. And so one of them, conventionally, neurologists don't do a lot of treatment for the gut.
No, well, that's down there. We're above all that, literally and figuratively.
Yes, that's why the liquid, the body fluid we're most comfortable with is cerebrospinal fluid.
Yeah, exactly right.
But we do in our clinic a lot of interventions that target gut health, and in fact, when we see in a child who's been progressing well, and we see agitation or dysregulation or an increase in repetitive behaviors, all of our staff, even our behavior therapists, our occupational therapists, immediately think gut. And so then as a team, we talk with the parents, we find out what's been happening, what the child's been eating, what their bowel movements have been like, what are the supplements are they on? Are they on things that may help, or have they recently stopped their digestive enzymes and now symptoms have emerged? And so what do we learn? So it's very much integrated into what we do. And I will say our approach has tended to be more symptom driven. And
so what would you do for gut treatments, for example, in these kids?
We use a lot of prebiotics and probiotics. I know that probiotics are, what we're able to give in terms of probiotics has probably less to do with the diversity of the healthy gut and more to do with what sort of species are able to be packaged and taken. So there's some interesting research that I've been following by Jim Adams at Arizona State University, where it seems that autism is highly associated with a reduction of gut microbiome diversity, and that because the gut has probably close to 1,000 different species of bacteria, what we're able to do with probiotics is
drop in the bucket.
Yeah, it's relatively small.
They're doing fecal transplants.
Yes, where it's in a sense sort of a super probiotic, where they're able to transplant close to 1,000 species rather than just 5 or 10 that are in a probiotic. So, but we use them and we see benefits.
What are they seeing with those fecal transplant studies?
The study that was published this year showed improvement. It was an 8-week course of therapy, so improvement in autism symptoms and GI symptoms during that period, and also 2 months following the end of treatment. And I've heard that there's some long-term data about to be published as well.
Like mild improvements or dramatic improvements?
Some with mild and some with dramatic, so it's sort of a range. Yeah,
it seems like a crazy therapy for autism, like fecal transplants, but if you're targeting some of the etiologies, because then that's going to have downstream effects on mitochondria, oxidative stress, inflammation.
Right. So it sounds unusual, but when you actually look at the mechanisms, there's a lot of rationale to it.
So there's a microbiome mitochondrial connection.
Yes, for sure. Everything's interconnected within the body and outside of the body.
Exactly, Amazing. And then how about the toxic load that these kids have? How do you, other than reducing exposures through organic food and so forth, how do you deal with, like, do you check for heavy metals? Do you
our testing for heavy metals is primarily for lead, and our approach is largely about trying to gently enhance the body's own mechanisms for detoxification through steps to increase glutathione. So we use a lot of N-acetylcysteine, a lot of antioxidants. That's been our primary mode of therapy.
I mean, I'm an N of 1 practitioner, but I think it's a shared experience among us doing this, that if you test for heavy metals in these kids, that they're often just loaded with mercury and lead on a challenge test, which you wouldn't see on a blood test, and they often really hold on to these because they aren't able to mobilize them because of low glutathione levels and oxidative stress. So it's really, if I had like 3 things to take with me to a desert island for autism, would be, gluten, dairy-free diet, antifungals, and heavy metal treatment.
Do you do challenge protocols here?
We do. We do. And we see it. I mean, across the spectrum of, neurologic diseases, we have a whole cohort of special forces soldiers here who are heavily exposed to blast training. They have indoor blast rooms where they blow things up and they shoot things, and they have extraordinary high levels of lead and metals. And it's fascinating how I saw a little girl with autism a couple of days ago, and her post-challenge lead level was 42, which is pretty high after a DMSA or challenge. And the question was like, where did she get all that? Because she wasn't eating paint chips and running behind cars in Cuba, with lead gas. And, so it's fascinating to see. And then those are, I think, like you said, those are really significant mitochondrial disruptors. Right. Yeah. And also, you wonder the regulation of antioxidant enzymes, because antioxidants we can take are a drop in the bucket compared to our own endogenous systems of catalase and superoxide dismutase and glutathione peroxidase and other inherent antioxidant enzymes. They're part of mitochondrial protective mechanisms. How do you upregulate those? Have you had any experience or thought about that?
I would just say that, and there's a slide I sometimes include in my presentations, which is I show the 2-dimensional representation of all of the biochemical pathways in the body. And it's extremely complicated.
Mind-blowing.
And of course, it's an oversimplification. And
it's not linear as it's mapped out.
That's
it's
right.
All, like it's a soup.
Right. And so does it make a lot of sense to work on a single pathway? Sometimes, and sometimes it's very helpful.
Yeah.
But the things that influence and shift that whole complex soup are what we eat and how we live and our quality of sleep and the quality of our relationships. So
this is great. I mean, it's amazing. And your work, is obviously focused on far more than the biological aspects. You're having doorways into these kids' brains through using music and other sort of modalities that allow them to sort of repattern what's happening.
I like to say the brain, especially the developing brain, can be influenced in so many ways, and that's an opportunity. It makes our work harder, and there is a real tendency in medicine these days to specialize because it makes something very unwieldy more manageable.
Yeah.
But I think it doesn't do a service to our patients.
Well, it doesn't represent reality of how our bodies are designed or organized, right?
And so, I think it's important to provide opportunities and to encourage trainees to be broad and to remain broad. And that's, I know, a part of functional medicine, which is to say, it's there's a way of looking at a whole host of diseases that conventionally are thought of as, oh, that's an autoimmune disease, that's a neurological disease, and it's not that way.
No, it's not. And what's fascinating is when you look at sort of extremes, for example, autism and Alzheimer's.
Yeah.
It's astounding how similar the patterns are in these. We look at a sort of a subset of SNPs, like we look at glutathione SNPs, methylation SNPs, and so forth, and you see that they share these, right? And then you see that they all have high levels of inflammation and oxidative stress and toxic load, and often the same therapies you're using, like the mitochondrial therapies in neurodegenerative disease, and then anti-inflammatory therapies and diet, and it's just fascinating. They're using ketogenic diets for autism and Alzheimer's as interventions. It's like, Well, I think we're pretty much really, at a new era of medicine. And you're a pioneer in that, and you're looking at sort of remapping our view of one of the most frightening and burdensome diseases that exist, that destroy families and that burden our whole healthcare system for the life of these children, and to be able to have an opportunity to start to reverse and stop these conditions is just amazing. So thank you for your work and for joining us today in Cleveland, and thank you everybody for watching, and hopefully we'll continue this conversation.
I hope so. Thank you so much.